Okay, so this is going to give you some more background information about some of the fungi that we're going to be talking about. We already mentioned Eucelago matus, and the other fungus that will come up later in discussions is called Microbacterium violaceum. Together, these two fungi are called smut fungi. They're part of a group of fungi called smut fungi. In this instance, Microbacterium violaceum is being illustrated during its infection process. This is a wildflower species called Sadlini latifolia. And instead of finding the pollen here, which you'd normally find in a flower of Sadlini latifolia, you've got the spores of the fungus that have replaced the pollen in the anthers. So this fungus is referred to as the anther smut because it infects the male flowers or the male parts of the species it infects. This illustrates what happens when there's an infection of Eucelago matus. Eucelago matus is able to infect maize or corn. It's also able to infect the thought to be ancestor of corn called teosinte. And it's able to produce these tumors or galls on any of the above ground parts, so any of the green surfaces. So the leaves, the stem, as you'll see on some other slides, also the tassels and the corn cobs. And it produces those tumors in which the spores of the fungus develop. So I'm going to do this by here. Okay, if you want. Yeah, so next slide. Okay, so these are some more images of Eucelago matus infection, if you want to think of it as cornography, since you're talking about corn smut. Again, showing infection not only of the stem and producing tumors, but also these tumors that are produced in the kernels. Now, I don't know how many of you want to eat something like that when you buy your ear of corn, and so that is actually a problem for America's farmers, if you would. But if by America you mean the United States, because a lot of people don't necessarily want to buy a corn on the cob that has that growing on it. And that results in about 2% loss per year, which doesn't sound like that much, but if you think about it, that corn is the largest legal agricultural product that we produce. That turns out to be about $200 million lost per year. So the farmers, they don't really like this, but I think it's all a matter of perspective, because if you see on the next slide, one person's pest is another person's delicacy. In Mexico, they actually intentionally produce this product for a dish that's called huitlacoche, which is it's found all over Mexico. You can buy it fresh in the markets like in Mexico City. And as I said, they actually have fields of corn that they intentionally infect. They do these focus groups with people. They say, well, does it taste better with this variety or that variety? Is it too gritty? And so they really try to optimize it, and you can get it in a can, and it's available in some of the Latin American grocery stores in town, and you can also buy it in some restaurants in town as well. So Mayan Cafe, if you've been there, there's a salmon dish that's got a sauce on it, and they describe as Mexican truffle what you find in that sauce. I guess if they said corn smut, they might not get as many takers. Also, there's a place called Guacamole off of Hurstbourne Road, north of Shelbyville Road, and you can ask for enchiladas or various other dishes with this in it. So, we have the next slide. One thing I want to point out is I normally give extra credit to people if they try this as a food, but I'm not sure if that's going to be an option in the near future. There are recipes online. If the Mexican or Latino grocery stores are open, there are some places that sell it in cans and jars, and so you can try it that way. But you can't really go to the restaurants. I don't think you're able to get any of this stuff. Although I did hear that Mayan Cafe is going to be doing takeout. So if you want to support your local restaurants and you can afford it, do takeout. Meanwhile, this is giving you another opportunity to see the life cycle of both of these fungi. We talked about these budding yeast-like cells, which are called sporidia, that are haploid, And if they're of opposite mating type, and we already said last time when we were talking about mating type, that means they have to be different at the A locus and the B locus. And if that happens, they will conjugate and form this connection. And again, assuming that the B locus is also compatible, then they'll produce this infectious dikaryon. Now, this infectious dikaryon actually produces a structure at its tip called an aprosorium that physically penetrates the tissue of the plant. In the case of these fungi, we think that that physical penetration has to do with enzymes that are secreted at the tip that degrade a bit of the tissue, allowing penetration. Now, inside the plant, that dikaryon will either migrate to the flowers in the case of the anther-smut, or will increase in biomass within the tumor that's developing. And then eventually, when the tumor is of the proper size for Eucelogomatus, the dikaryon will have the nuclei fuse and produce these diploid structures called teleospores. And then those teleospores are what gets released from the burst open tumor. And once a teleospore lands on a suitable surface, it can germinate and undergo meiosis to produce more of these haploid cells and basically repeat the process. Okay? So all that's pretty cool, right? Yeah, go smut. What we're going to move into next is, we've talked about the genes involved. Now we're going to move into talking about signaling pathways that allow this to take place. All right, this is a reminder of what we talked about before with the A locus and the B locus. So the A locus has these two genes that are important: the MFA locus coding for the pheromone, and the PRA locus, PRA gene coding for the pheromone. And the same thing is true in the other mating type A2. So you encode a pheromone of your particular type, and you encode the receptor for the opposite pheromone. And so that interaction, that signaling, actually affects a transcription factor that we'll talk about later called PRF1, and that transcription factor will interact with the B locus, activating those genes, and then eventually you get the heterodimeric transcription factor that's compatible. So B East 1 and B West 2 is an example. And then when that transcription factor is formed, you get sexual and pathogenic development. Okay? And then next slide, and then next slide. Okay, now, a number of you probably been exposed to signaling pathways before. Yes? Yes. Did you enjoy that? No, not really. Well, a number of you, your blood pressure goes up, and stress hormones start coming out when we start talking about signaling. So this is going to kind of be a general reminder of what signaling components there are. And then what we'll do is, in the next gathering or the next recording that we do, we'll talk about the specifics of components of those pathways. So first, just again, to make it so your blood pressure doesn't go up, we have components called G proteins. So G proteins are GTP binding or GTPase proteins, and there are different types. So there are what are called heterotrimeric G proteins, in which case you have 3 different subunits, and you have an inactive form of the G protein when they're all together, and you have GDP bound to one of the subunits. That becomes active when GDP is exchanged with GTP, and that allows for the separation of, in this case, the alpha subunit from the beta and gamma subunits. There are also other kinds of small GTP binding proteins that work in a similar fashion. So Rho is an example, and this G protein is inactive when GDP is bound, but when a guanine nucleotide exchange factor, so basically this exchanges GDP for GTP, then that's the active form of Rho. And there's also proteins, these GTPase activating proteins, that can make the switch back this way. One thing that people often get confused about is this is not simply a matter of phosphorylating the GDP. What the exchange factor does is it physically removes the GDP and replaces it with a GTP to activate the protein. Okay, so that's G proteins. Any question about G proteins, just the overview. Okay, next. All right. So the next thing that you may have heard of is something called the MAP kinase pathway. So does everybody know what a kinase is? What's a kinase?
Phosphorylates.
Phosphorylate something. Well, MAP kinases are protein kinases. So the name MAP kinase stands for mitogen activated protein kinase. And so what that means is these are signaling pathways that contain a cascade of these different kinases. And what they do is they phosphorylate specific amino acids on their target proteins. So specifically, these proteins phosphorylate serines and threonines in their target proteins. And typically there's a cascade that involves what's called a MAP kinase that phosphorylates the MAP kinase, which in turn phosphorylates the MAP kinase. And then the MAP kinase typically is going to phosphorylate a transcription factor, and that's going to change gene expression in response to a signal. Okay? So that's the MAP kinase pathway. And again, these are overviews, so you don't feel too freaked out when you start to see the details of some of the pathways we're going to discuss. So, the next slide. So the other type of signaling pathway that we will talk about is something called the cyclic AMP dependent protein kinase A pathway, or PKA pathway. And PKA is usually composed of a regulatory set of subunits that inhibits the catalytic subunit. And it turns out that the regulatory protein, the regulatory subunit can be bound by Anyway, regulatory subunit that's blocking the catalytic subunit can be bound by cyclic AMP. And cyclic AMP is synthesized by adenylate cyclase, and when cyclic AMP binds to the regulatory protein, it releases the regulatory subunit of the complex, and it leaves the catalytic subunit active, and then the catalytic subunit then can phosphorylate targets which include transcription factors. And again, what that leads to is a change in gene expression as a response to a signal. Okay? I think the next slide is actually that. Okay, so that's the end of these slides, and then we will continue on the whiteboard in just a few minutes.